Hello everyone, and a warm welcome to all of you for this online lecture on photosynthesis, which is an important topic for your NEET examination. Now if you look at the previous question papers of NEET, you will find that there is a trend of asking questions on scientists and their contribution in the field of photosynthesis. So keeping this in mind, we'll just go through a list of few scientists and their contribution in the field of photosynthesis. So the first among them is Joseph Priestley. He demonstrated that the green plants purify foul air and convert it into pure air. The second one is Jan Ingenhousz. He demonstrated that photosynthesis takes place in the presence of sunlight. That means sunlight is essential for photosynthesis, and he demonstrated it through various experiments. The third scientist is Julius von Sachs. He proved that chloroplast is the seat of photosynthesis, which means photosynthesis takes place in chloroplast with the help of sunlight and chlorophyll. Now the fourth scientist is Cornelius van Niel. He demonstrated that some bacteria use H2S, that is hydrogen sulfide, instead of H2O in the process of photosynthesis. So this was regarding a few scientists. Now we go to the photosynthetic pigments. Now what are pigments? Pigments are the substances that have an ability to absorb light at specific wavelengths. The color of leaves is due to four pigments: chlorophyll A, blue-green color; chlorophyll B, yellow-green; xanthophylls have a yellow color; and carotene, yellow to yellow-orange color. Since chlorophyll is the most important element for photosynthesis, let us have a look at the structure of chlorophyll. Chlorophyll has a tadpole-like structure. It consists of a porphyrin head and a phytol tail. So this diagram shows the structure of chlorophyll. This is the porphyrin head and this is the phytol tail. Now chlorophyll A is the primary photosynthetic pigment. All pigments other than chlorophyll A are called as accessory pigments. Now, out of all the pigments, chlorophyll A is said to be an essential pigment, or it is said to be the primary photosynthetic pigment. Now, why is it said so? Because only chlorophyll A has the ability to transform solar energy into the chemical energy. That is, it can convert solar energy into ATP. While the other accessory pigments, they have the ability only to transform the absorbed energy to chlorophyll A. Accessory pigments cannot transform solar energy into chemical energy. So therefore, chlorophyll A is called as the primary photosynthetic pigment. And the accessory pigments are also called as the antenna molecules. Chlorophyll B accounts for about 1/4 of the total chlorophyll content. Now the another type of pigments, which are carotenoids, they protect the plants from excess heat and prevent photooxidation of chlorophyll pigments. Thus, they are also called as shield pigments. So in this portion, we have to remember 3 points. The first one is that chlorophyll A is called as the essential pigment. Then accessory pigments are also called as antenna molecules, and carotenoids are called as shield pigments. Now, let us have a look at the chloroplast. I assume that all of you know the structure of the chloroplast. So let us just see a few points regarding chloroplast which are important for your examination. Now chloroplast is the seat of photosynthesis, which means photosynthesis is carried out inside the chloroplast in the presence of chlorophyll and sunlight. Now these chloroplasts, they align themselves along the walls of the mesophyll cells. These chloroplasts align themselves along the walls of the mesophyll cells. Now why do they do so? This is for easy diffusion of the gases and to receive optimum quantity of the incident light. So for these two main reasons, the chloroplasts align themselves along the walls of the mesophyll cells. Now the second point about chloroplast is that they align themselves in the vertical position along the lateral walls of the mesophyll cells. How? Why do they align themselves in the vertical position? The main reason is to protect themselves against the destruction by light. They protect themselves against the high light intensity. High light intensity can destroy chloroplast, therefore they align themselves in the vertical position to prevent this destruction by light. Now another important topic under photosynthesis is chemiosmotic hypothesis. This chemiosmotic hypothesis was explained by P. Michel. This mechanism explains how ATP is synthesized in the chloroplast. Now the diagram which you see over here is the figure of the lumen of the thylakoid. This is the thylakoid and this portion is the lumen of the thylakoid. Now all of us know that during photolysis, water is broken down into hydrogen and oxygen. All those hydrogen ions or the protons, they get accumulated in this lumen of the thylakoid, and since the number goes on increasing, they build up a concentration gradient in the lumen of the thylakoid. Now this structure which you see at the membrane of the thylakoid is nothing but the enzyme ATP synthetase. Now when the concentration increases, when the concentration of the hydrogen ions increases, the synthesis of ATP becomes easy with the help of this enzyme ATP synthetase. Now this enzyme ATP synthetase has two parts, F0 and F1. F0 forms a transmembrane through which H ions pass from the lumen to the stroma, thus breaking the concentration gradient. So these hydrogen ions, they pass through this transmembrane. They pass through the F0 portion of this ATP synthase and they come out into the stroma. So because of this, the concentration gradient is broken down. And the second part of this enzyme ATP synthetase is F1. F1 is the portion that protrudes on the outer surface of the thylakoid. Now the breakdown of the gradient provides enough energy to cause a change in the F1 part of the ATPase, which makes the enzyme synthesize several molecules of ATP. Now this is how ATP molecules are synthesized, and the energy for the synthesis of ATP is acquired by breaking of the concentration gradient. Okay, so now we turn to a next small topic, that is emergence enhancement effect. Now let us look what is this effect all about. Now let us look at another small topic which is called as the emergence enhancement effect. Now let us look at what is this effect all about. The simultaneous application of light of a shorter and a longer wavelength increases the rate of photosynthesis. This means if a plant is receiving the light of a single wavelength, then the rate of photosynthesis does not increase. But if you apply light of a shorter wavelength and a longer wavelength simultaneously to the plant, then the rate of photosynthesis is enhanced. That is why it is called as the enhancement effect, because it enhances or increases the rate of photosynthesis. And this was discovered by the scientist Emergence. That is why it is known as emergence enhancement effect. Now the second point regarding this topic is that when monochromatic light of more than 680 nanometer wavelength, that is the red region of the spectrum, was used alone, it reduced the rate of photosynthesis. Which means if you use only one light, if you expose the plant to only one light of a single color, so for example let us take red color which is of a wavelength 680 nanometers. If the plant is continuously exposed to this wavelength, then there is a drop in the rate of photosynthesis, which is also called as the red drop. So once again, when you apply light in a simultaneous manner, a light of shorter and a longer wavelength, then only the rate of photosynthesis increases. So this is emergence enhancement effect.